what we'll do is I'm going to open chat, and why don't anyone who knows what they would like to do for the review, put it into chat like we did last time. If you have specific issues, like, make sure, if you just say essay questions, that's too much to do in the time we have, but tell me specific essay questions. I'm happy, do those essay questions. So the sample exam or just open questions, a lot of people had some good questions in the review, the office hour, so why don't you think about what you guys want and put it in the chat. Let me get my essays now that I should have them open just to make the concept of glucose 6-phosphate. Great. Okay. Let me go into the essays. Essays. Okay, close this thing. Sample exam 4. Okay. Push this off to the side. Close that. Too many things on my desktop here. Where are the sample exams? So there they are. I moved them, that's why. Okay, so far I'm getting 2 things. Sample exam, sample final, midterm. Here we go. Okay. Okay. So because Kaylin's is kind of brief, we could start with that one. Let me open this up. And anyone coming in, what we're doing is we are putting into chat what your questions are, what you want me to review. And let me get chat open again. Oh, I'm almost confused where it shows up here. Here it is. Okay, more chat. Okay, let me put chat off to the side here. All right, so Kaylin asked about glucose 6-phosphate. So if we are in any cell in the body, so I'll put any cell here, and there's glucose in the blood, and this is its plasma membrane. The glucose is typically co-transported in any cell in the body with sodium because of the secondary active transport. And as soon as the glucose gets in the cytoplasm, there's an enzyme that's the first step in glycolysis that converts this, and I think it's called glucose. I don't remember what it's called, probably glucose phosphatase or something like that, but it turns it into glucose 6-phosphate. So as far as the body is concerned, the cell is concerned, this is always on a downward gradient, concentration gradient. So you always are seeing higher amounts of glucose out in the blood because there's basically 0 glucose in the cytoplasm. Is that what you're looking for, Kaylin? You can just say things if you have. Well, anyway, the only other thing I'd say about this is then this gets a choice. Glucose 6-phosphate could go in anabolic pathways and become glycogen in the cell, or it could go catabolic pathways and become ATP by going through glycolysis and the Krebs cycle. So the only unique thing is if you are not any cell in the body, if now you are a liver cell, the PCT cell, or the absorptive cell of the duodenum, of the small intestine, mostly the duodenum, then you have the ability to take glucose 6-phosphate and turn it back into glucose and, God, I can't write, can I? Glucose, and that's the glucose 6-phosphatase, which I said you don't have to name the enzyme, but you have a unique enzyme that allows you to take glucose. Now that glucose could come from glycogen that is through glycogenolysis, and that could be the source, or it could be gluconeogenesis, where you're taking fats and proteins and running your Krebs cycle and glycolysis backwards to convert those carbons and hydrogens in those molecules into glucose and then secrete it into the blood. So hopefully this is what you're looking for. One last thing, just because we are reviewing, is there is a hormone, and the hormone that allows this to happen is insulin in most cells. However, not required for the liver or your brain. For that process, I'll put or your CNS neurons. They don't require insulin to do that process either. And PCT, as far as I know, doesn't require it either. That wouldn't make sense. And the absorptive cell probably doesn't. But the only one I think the only one the book says is the liver doesn't require insulin to take it up, because insulin has a different effect in the liver. What insulin's going to do, Jesus, I don't know why they put that up and down on this program, right, where right-handed people would put their hand. But anyway, insulin is going to make this process and this process increase. So you're going to turn more glucose into glycogen, and you're going to suppress this process where glucose is turned in. That's encouraged by glucagon, but this process is encouraged by glucagon. So if we change colors again, so glucagon would be the one that would increase this process and this process, and glucose increases the other way. Is this way more than you wanted, Caitlin? I don't know. She's probably here, and
this is good. I wrote it in the chat.
Good. Okay. All right. So the other requests I'm getting is sample 4, the sample exam, and then could we review essay 5, functionalities of the liver, looking at lipolysis and protein synthesis. We can do that. And then there's just a general question. So I think the liver one would be a good one to do if you really want to just focus on lipolysis and protein synthesis. We might want to do that now. If you want to more do the all of the functions of the liver. It's good for a review. We could kind of touch on that during review. But since we're in all this metabolism, maybe I'll do that first. In fact, during the office hour, Sana brought up, or maybe it was Jessica, I think it was Sana, an issue where I hadn't really looked at this or thought about it for the figures on the protein. Let me get there so I can tell you which figure it is. Okay. It's in chapter 24, and the figure that's embedded in the slides as well about protein processes. It's figure 2417. 2417. And the title of the figure says, processes that occur when amino acids are utilized for energy. And the particular thing it's actually showing should actually be labeled catabolic processes, because I took the time to look up glutamic acid that is the product of transamination in that figure. So if you're struggling to get to the figure, I'll give you a second or two. And I was looking at that, always assuming that glutamic acid was an essential amino acid that was being made. And that's not the case. We looked up the list of essential amino acids, and that's not one of them. It also showed, when I just briefly Googled it, that there are multiple pathways that can lead to that transamination occurs with, not just this particular one that they called the ketogenic pathway. So I think this figure is showing transamination so that you can do gluconeogenesis. So it's not trying to show you pathways for gluconeogenesis is really what it should be titled. And if you go over and you look at the text as well on the next page, it's the heading is degradation of amino acids. So it's really talking about breaking down amino acids to do either gluconeogenesis or to make ATP. So there is transamination, however, which is anabolic, and it's just not shown in that figure. So its definition is straightforward. It's the conversion of one amino acid into another amino acid. So you're taking a non-essential amino acid, could be the case, and you're making it into another amino acid. Now that amino acid could be essential or it could be non-essential. If it's essential, you might be making it for anabolic reasons in order to generate that amino acid and release it into the blood for other body cells. And the liver does that. The particular picture is showing it for catabolic reasons, where it's making it into a non-essential amino acid that then is broken down like they're showing with oxidative deamination and keto acid modification in order to insert it into the Krebs cycle, make ATP out of it. So I don't know if that's what you were asking in that, but because it came up during the office hour, and it was something I never thought about, then that figure really is kind of mislabeled in a way, and it's really only focusing on this, making it into a non-essential, and that could then be made into ATP, or it could be made into glucose. Is that making?
I'm sort of confused, Professor. Does that mean we can take non-essential amino acids and make essential amino acids? I thought the whole point was we couldn't.
I went the other way around. You're right. No wonder. Non-essential. You're right. You could make any amino acid into, yes, can make any amino acid into any. You're right, I misspoke. I switched the essential and non-essential. You got to eat essential amino acids. They have to be in your diet, and your liver isn't making them. You're just making another amino acid into another non-essential by transamination along anabolic pathways. Sorry. Got to wake up this morning.
And I'm assuming that the amino acid that gets made is just dependent on what your body needs.
Yeah, and I don't know what regulates that.
Right. Okay.
Okay, and then lipolysis. What specific part of lipolysis are you worrying about, Noah?
Well, the whole like what is I guess important there, because I understand triglycerides can go into fatty acids or glycerol, and then the important thing about fatty acids is beta oxidation, but glycerol can turn into glycogen. Is that what it is? And that's how that whole process works, different from there.
Okay, let me get to the figure.
It's just confusing, the figure, to me, just a little bit with all the arrows everywhere.
24, which one?
Let me double check. It's 2416.
Okay, I'm going the wrong direction here. 24.
That's what it says on the slides.
You know, this is almost like a figure just showing you an overview of both anabolic, and anabolic would be pink, and catabolic pathways. So if what they're saying is if any cell makes a ketone body and releases it into the blood, the liver is able to take it out of the blood, converted it into acetyl-CoA, and make it go into Krebs cycle, make ATP, or it can do gluconeogenesis. Let's say you're making glucose from a fat and you're a liver cell, then, or a adipose cell, it says, then you could take a triglyceride and you can use lipolysis, which is the blue arrows, to break it down to glycerol and fatty acids. The glycerol is inserted into the Krebs cycle someplace, they don't say where, or into glycolysis, and that is converted into glucose, probably combining it with some intermediary. And then the fatty acids, through beta oxidation, become acetyl-CoA. Now that acetyl-CoA, if you're trying to do gluconeogenesis, you can actually convert the acetyl-CoA back into pyruvic acid, but you're going to generate bile salts. You could also be doing it for generating bile salts, acetyl-CoA it's showing. So you can take that fatty acid and convert it to acetyl-CoA and make it into bile salts. You could make it into ATP. So you've kind of got some choices, and they're trying to show all those choices. Is that what you're finding confusing, Noah?
Yeah, it just seems like a broad overview, but I mean, I get like the face value of it. The triglycerides mainly, are those the chylomicrons that they would be talking about, or?
Well, the vast majority of the fat that's in chylomicrons are triglycerides.
Right.
But as the book does tell you, some monoglycerides are absorbed directly into the blood and don't have to be in a chylomicron, and they can make it to the liver more rapidly that way. So I don't know what makes the difference, why some monoglycerides end up being just directly absorbed and into capillaries. They must be small enough or something to do that, whereas others are taken up, put into chylomicrons, and those end up being some of the triglycerides. But you could be storing triglycerides in the liver or the adipose and going through this pathway too, because the liver stores quite a bit of adipose tissue. Is that what you're asking?
Yeah, yeah, that makes sense. Thank you.
Okay, so So the next thing up there has to do, let me just type something in. Looks like someone else is making a request. Professor, please explain the principles of ESC. Okay, so that one's back more in the review part of the cardiovascular system. So let's do sample 4. Let's stop sharing this and open it up. And then we'll share that screen. And I don't remember how old this test is since I've, this is the PDF version of it, and I took that off of it. So it could be quite old. And so we'll see whether it matches the stress that I've made this semester on it. So walking through is this what, I guess I won't, let me get that chat back up since I should have that up too in case you guys put a question in it. There you go. Get it to show up again. Okay. All right, so let's walk through this. I'll have chat open if you have something. Otherwise, you can just shout it out. So the first one is, what is an immune type cell? Well, clearly Peyer's patches is part of MALT. So there are B cells. I'm going to try and use this also for a little bit of the comprehensive review when needed. So in those Peyer's patches, remember, you're going to have B cells awaiting their antigen. You're going to have T cells that are CD4 cells. You may have CD8, but the main one you're going to need is CD4 to get the B cells activated. And you're going to have dendritic cells. So Peyer's patches are going to be able to trigger an adaptive immune response very rapidly by removing pathogens from interstitial fluids to have them encounter their B cell and their CD4 cell within the MALT system. I'd say goblet cells are, because what they're secreting is largely mucus, and that mucus is part of your barrier system, so it's trapping it. If it's past your stomach, or not in goblet cells in your stomach, which you don't have a whole lot of, what you're doing is trapping them and then expelling them in your feces. Parietal cells clearly make HCl, and the stomach is killing a lot of pathogens with HCl. Duodenal glands are making bicarbonate, so they're not, from my knowledge, they don't contain any lysozymes and stuff, although, I could be wrong about that. It doesn't mention them containing that. It's a bicarbonate-rich solution. So lysozyme is used to, the last one is used to battle gram-positive bacteria by digesting their cell wall. So the best answer here is the duodenal glands. Is this?
Professor, I had a question about that.
Okay.
On your slides, you put that the Brunner's glands also secrete mucus. So would that not be?
Brunner's are duodenal.
Yeah, you say that they secrete mucus.
Is that what the text says too?
I haven't checked the text. That's what your slides say.
Yeah.
And it says that it releases antimicrobial agents as well. So
it does.
That's what your slides say.
I probably have to bow to that. I don't remember mucus in there, although maybe the slide they are. Let me just see if the text says anything about. I find them. So again, I wrote this test a while ago, and I may have changed things as the text changed. So duodenal glands. Duodenal glands. Maybe they don't unfortunately have it in the, oh yes they do, 903, 902, 903 in the index. Two.
It just says it's an alkaline-rich mucus in the book.
Yeah, I see it here. So it does make mucus. It doesn't say anything about the antimicrobials, although it could. So, it's not the best question. So it's probably just something that's changed over my time. Clearly, I would say the Peyer's patch, the goblet cells and the parietal cells and the lysozyme. So, this is probably at some point it just said bicarbonate. And so that's why I just taught it as bicarbonate. Are we okay? There was another question. Somebody was speaking up. Was it the same thing?
Yeah, I was just also going to say
Same
that.
thing. Yeah, again, my guess, I usually put up questions or old tests from, 6 or more years ago. So it could be that it was a couple editions back and they didn't mention mucus and they just mentioned bicarbonate. And so that's how that ended up like that. And that's what I taught. Okay, so this one's, again, hopefully more straightforward than the last one. This is the layer right in that mucosal layer of muscles. So it can't cause movement of the whole alimentary canal like segmentation or peristalsis. It does make local movements for churning, and it does increase with gastrin. So gastrin causes all the smooth muscles to move more, and that's going to include the muscularis mucosa, so it's C and D. Any issues with that one? Okay, good. So saliva does contain mucin because that's the sticky stuff that makes mucus sticky. It aids in the digestion of proteins. No, it does not, right? No proteases in your saliva. It does help prevent cavities, which we didn't talk about quite a bit, although the book does, that if one is continually washing away the acids that are produced by the plaque-producing bacteria that erode your enamel on your teeth. So enamels, just for review, is a calcium salt, and like limestone, it's eroded by acid, and that's what's being made by these plaque bacteria. They're making a variety of acidic waste products, and that tends to erode the enamel and cause cavities. So saliva is, if nothing else, is just rinsing off the acids to keep them away from the enamel. And it is not an endocrine, it's an exocrine gland. Hopefully that's easy. So the answer is A and C. Which of the following does not help neutralize the chyme? So these are sources of bicarbonate. So lacteals are the lymphatic, so they're not going to be doing it. So the rest is duodenal glands, bicarbonate rich. Bicarbonate's added to bile as the liver makes it. Pancreatic juices are going to include the ductal cells secretions. So these guys are all going to help neutralize, but not the lacteals. Okay, which of the following would increase stomach motility and open the pyloric sphincter? So if the pH drops in the stomach, that means it's becoming more acidic. And if anything, eventually, if it becomes acidic enough, it will inhibit the contractions of the stomach. Remember that little negative feedback if it gets below 2. And the pH, as far as I know, pH doesn't stimulate. Stretch can stimulate motility, but as far as I know, pH doesn't. Secretin is going to suppress the stomach. It's one of those ones that feeds back and suppresses motility of the stomach. Parasympathetic this should be myoenteric. Again, this may have been a change in Yeah, it could be enteric. I think they're including both submucosal and myenteric because it's not actually here. Motility could be churning as well, but to really open the sphincter, you need the myenteric plexus. But I think that's fine. So that would, and then stretch stimulates that little local reflex as well as the vagovagal reflex that then increases more ACH release, and therefore you get it. So it's C and D. Are we good on this one? Am I going too fast? Okay. So if you don't have intrinsic factor, then it would lead to the vitamin B12 deficiency. Since intrinsic factor is released by parietal cells, this maybe is tempting because it would be a secondary effect, but really the question is directly saying, what does intrinsic factor do? So intrinsic factor allows you to absorb vitamin B12 by your small intestine. So it's one of the things that is absorbed in the jejunum a lot because it's fat soluble. So often you need to get it down further before it's actually active, but it's probably also absorbed in the duodenum. But I believe the book says the jejunum does quite a bit of vitamin B12. So an ability to absorb fat, it's not used directly to absorb fat. In cancer, it's just a wild question. And glucagon is not stimulated by vitamin B12. It is stimulated by low blood glucose levels. So it's not relevant here at all. Any questions on that one? Okay, so what's the name of the sphincter that allows bile in pancreatic? So we put all these sphincters here. Esophageal sphincter is what keeps it's also called the cardiac sphincter. I think they put it in the book as esophageogastric sphincter or something like that, but it used to be called the esophageal. At any rate, it's the cardiac. I tend to just call it cardiac. And that one is preventing the acids of the stomach from eroding the lining of the esophagus and also closing up the stomach as like a closed bag so that you can toss the food back and forth within it to do mechanical digestion. The OD is the one that I've been using that name mostly, but it's also in the book called the hepatopancreatic sphincter. So I probably would use both names if I had it in the test. Not just OD, but you saw it in the lab test. I just found it easier to type OD than that one. So pyloric regulates exiting from the stomach, and we could talk more about that one later, but it's closed and opens only when great deal of contractility happens in the stomach, and then opens slightly to allow a little chyme into the duodenum. Ileocencals between the ileum and the cecal, or the opening of the large intestine, and that one's closed to prevent bacteria from entering the small intestine, and it opens as gastrin and motilin levels increase contractility of the whole alimentary canal and allows a little bit of the chyme into the large intestine, in particular into the cecal area, where gravity would pull it down into the cecum and enable it to probably expose it to the large number of bacteria in the large intestine, and therefore my brain this morning. Appendix is right there, which is malt, and help educate your immune system on what kinds of bacteria are in your large intestine normally, but also if anything's made its way through the whole small intestine and enters the large intestine, you have some Peyer's patches in the ileum, but now you have your appendix right there, which is part of MALT, and there's crypts in the appendix that help educate your immune system to these new potential guys coming in. And the last one is the anal sphincter, which I hope is by default closed. And there are two of them. There's an internal sphincter and an external. So I didn't say which, but both of them at rest are contracting and closed. And the sympathetic is closing your internal, which is smooth muscle. And your somatic nervous system is chronically active to close the external. We good? Okay. Well, I was wondering if I left a question C. Do you could possibly move to Thursday? The reason I wanted the lecture questions due today is so that you could have some time to just focus on your comprehensive part. But if you turn it in Thursday, I won't take late points off. So people are asking about this. I mean, we're talking 1 point is probably what I would take off, but which isn't going to make a big difference in the long run. So you guys, if you really feel you need to have 2 more days to work on those lecture questions, then it's not what I'd recommend, but if you need it, I won't punish you over it. So which of the following retroperitoneal, this is right out of your lab test. So the duodenum, or the vast majority of it is, the ileum is not. The esophagus is not, because the vast majority of it isn't even in the peritoneal cavity. It's in the thoracic cavity or in your neck and oral cavity. So, and the little bit that's in the stomach is still within the peritoneal cavity, not behind it. Pancreas is retroperitoneal entirely. Hormone that causes the contraction of the gallbladder. So know those hormones. If we know it's CCK that does that. Gastrin does not, as far as I know, cause increased secretion or increased motility there, although it does along the alimentary canal. The only other thing that could be a right answer for the gallbladder would be ACH. So ACH rises even during the gastric phases and the cephalic phases, causing the gallbladder to do weak contractions. But once you get both ACH and CCK, which would rise when the chyme that's fatty reaches the duodenum, then you're going to get strong contractions. So the greater stimulus, or the strongest stimulus, is from CCK, and but acetylcholine would be a possible answer here too. So here, you have the argument that, oh, well, why isn't it secretin? Secretin is not targeting the smooth muscle itself. It does target the enteroendocrine cells that cause CCK then to be released, however. So these are a secondary effect of secretin is to cause CCK, and then CCK causing the gallbladder. But I would not accept secretin as an answer because it's not directly stimulating those smooth muscles. VIP actually causes relaxation of the alimentary canal's smooth muscle, but again, I don't think it affects the smooth muscle of the gallbladder, but it clearly wouldn't cause contraction of it. One of the things it does do, though, that's kind of unique is to cause vasodilation of the capillary beds, or of the arterioles feeding the capillary beds, so that you're better able to absorb nutrients. But as far as I know, it doesn't affect the gallbladder again. And histamine is only targeting the stomach here. And as far as I know, it only targets parietal cells, but I could be wrong about that. The only one we know of is it's increasing HCl secretion and probably intrinsic factor secretion by parietal cells.
Professor, I had a quick question about VIP.
Yeah.
What actually stimulates the secretion of VIP?
It's neural, so it's part of that plexes talking to each other, as far as I know. I don't know the more of the details of that.
It's just that it can also act as an enterogastrone, right?
Yeah, so it's they say that, but really all the parasympathetic and sympathetic kind of release generally into the interstitial fluids. So in that way, it's an enterogastrone. I mean, you're saying a hormone, basically, right?
Right.
Yeah. I mean, I would argue all of the neurotransmitters, since there's no real neuromuscular junction, they're just kind of releasing it locally. But the nerves are bringing it closer to the muscles to cause the vasodilation, and the muscles of the intestines in order to cause them to relax. So clearly there must be an excitatory on one and an inhibitory. No, I guess they'd both be inhibitory, right? They both be inhibiting smooth muscle. But what else I think VIP does some other things too, right? Increases secretion. It doesn't say of what, but it inhibits the parietal cells. So there are some excitations. Now, whether that's in the pancreas, it's what it's secreting or not, it's not telling us. So it does seem to have both stimulatory effects in the pancreas and the small intestine, but again, they're not saying what. So I wouldn't ask the things that are vague like that, if that's worrisome to you. I would focus for VIP on the vasodilation part and maybe the relaxing the intestinal smooth muscle part, and inhibiting the stomach's acid. The other two are kind of vague. Stimulates buffer secretion, maybe it isn't all that vague. And then it just says pancreas increases secretion.
I also had a question about the relaxing of the intestinal muscle. Wouldn't you want them contract to like move it along through peristalsis and whatnot? Like, why would you want it to relax? Is it to have the pancreatic juices act on it, or like whatever?
And maybe that tension between needing things to stay long enough to have things act on them, like you're saying, and at the same time moving things along. So maybe there's some balance that's being made between the two. Unfortunately, the book doesn't have a lot to say, if I remember right, about VIP. So, yeah. Okay, beta cells. This is just what do they make? So the secrete a watery bicarbonate rich, that's the ductal cells, right? Release glucagon when blood sugar is rising. Well, none of that makes sense. Glucagon is secreted when blood sugar is low, and it's from alpha cells. Insulin when blood sugar is rising. That's true, even though it's misspelled. So what other things can stimulate insulin besides, can stimulate these beta cells? So the arrival of fatty chyme in the duodenum gets the release of GIP that can stimulate it. And there are other stimuli, but I would focus for insulin on the digestive ones for now. I'm sure in the lecture I talked about it more, that some of the other hormones that are released, if I remember right, the somatostatin, maybe they have that one in here too. No, it's inhibited, if anything, by somatostatin, I think. But, I'd focus, those two are the only ones I'd worry about, the GIP and the rising blood sugar levels. There are other stimuli for insulin release, however, but let's just focus on the digestive one. So trypsin and other hydrolytic enzymes, those are the acinar cells, and they're these guys are exocrine, as well as the ductal. The other two that make insulin and glucagon are endocrine. Stimulated by secretin. Secretin stimulates the ductal cells, not the beta cells. Hopefully that's good for everyone, but gets us to touch on some of the other things. And then chylomicrons, they cross the luminal membrane into the absorptive cell by simple diffusion. They're made in the absorptive cell, so they don't cross the luminal this is old enough, this is apical. So the older editions of the book called this the luminal membrane against the lumen, but now they're calling this apical, if that was the confusing part for you. So what crosses are the monoglycerides, diglycerides, and the fatty acids by simple diffusion into the absorptive cell, not the chylomicrons. Transport cholesterol from the liver to the adipose tissue. So these are the VLDLs. So if you're confused where I'm going with that, let's see if I can find that figure. We're looking at figure 24.4, and this is basically testing that figure. So VLDLs, it says, carries triglycerides from the liver, mostly to adipose tissue. That's these guys, are primarily made of hydrophobic proteins. Well, hydrophobic should be a red flag. They should be amphipathic proteins, right? Because you need to have some hydrophobic part to interact with the fat and some hydrophilic part to interact with the water. So even though there is quite a bit of protein in chylomicrons, it's one of the lowest amounts of protein of the lipoproteins that are in the transporting this fat, but the hydrophobic right away is wrong. Are transported by lacteals. That is true. Once they undergo exocytosis out of the absorptive cell, the lacteals pick them up and they get milked as those muscularis mucosa causes the villi to go up and down, moving the chylomicrons into the lymph that then ultimately end up in your blood. They're not made by the liver. There are ones made by the liver, but not those. VLDLs are made by the liver, for example. We all right? GIP stimulates insulin. Yeah, so that was probably from an earlier question. Okay. Secretin release is stimulated by acidic chyme, and that ideally should say in the duodenum. So bile release does not directly stimulate anything. Bile manufacturing is stimulated by secretin. Release, if anything, is probably from the gallbladder, CCK. So once again, it's secretin, even though it causes CCK release, that's a step back. So I wouldn't accept that. And then gastrin, as I mentioned, increases motility along the alimentary canal, but I don't believe it, according to the book, it doesn't mention at least that gastrin is causing gallbladder activity to go up. Secretin, and certainly doesn't cause secretin to be released. And then carbohydrates in the chyme, don't seem to stimulate any particular hormone specifically. So carbohydrates in the chyme per se don't seem to be detected. Fats in the chyme would be CCK or GIP that are stimulating those. So what you might want to do with that hormone table is maybe make just rearrange it. I'm a big advocate for studying when you have a lot of information like that. Maybe taking the activities by who, like the pancreas, and then all of the various things that are stimulating a hormone to be released, and then how that hormone acts in the pancreas. So here they put they have the targets all jumbled together. Maybe make it a new concept map by having, okay, here's a target. What's all the things that could talk to that target? And that would be one way of trying to get that table studied down. Okay, just a suggestion, up to you. But I like.
A quick question, Professor.
CCK does stimulate secretin and vice versa, correct?
Yes, but it's a step back. So.
Right. This is obviously a case of the best answer.
Yeah.
Okay.
Well, and even in this one, I wouldn't accept CCK because this is saying what stimulates. Oh, I see. I would accept it if it was one of those. Yeah. No, but it's, yeah. So if there was a CCK on here as one of these five, I would accept that. It would be both the acidic chyme and the duodenum. You're right. Okay. This is what an earlier request for the for what are the roles of the liver, and that is essay, 4 or 5? Of course, I have them open, but they're in another sheet. So essay 5, describe critical roles the liver plays in maintaining storage and metabolism of all these things. The liver also plays biosynthetic roles. Describe the context in which these roles are played, immune context, but this is really a good review question. But this is just taking the concept map or the table in the book and telling me one of them. I can't write on this, but perhaps I should have transferred it over to my tablet. But it stores lots of stuff. So vitamins, vitamin A, vitamin B12, vitamin D. It stores largely fat-soluble vitamins because it's also storing fat. One of the, I guess, more rising numbers of pathologies is people who get fatty liver, where their obesity has led their liver to become so fatty that it interferes with the functioning of the liver itself. So it's storing that fat, it's storing glycogen. Fat and glycogen, it's storing because it's the pantry of the body to maintain blood glucose levels so that it can use glycogenolysis and glyconeogenesis to cause the fat or the glycogen to be converted back into glucose and secreted into the blood. It's going to do that under the influence of glucagon plus other hormones like epinephrine and corticosteroids and potentially growth hormone. You can, again, look at that table on the... So what else does it store? It stores minerals, particularly the one we focused on is iron. It's storing it on ferritin largely, and if it has enough iron, it'll store it on hemosiderin. Other storage things, maybe I should get the table out so I have it somewhere. Mine myself. That's about all they have in the storage part in that table. So we went through lipid metabolism. It stores a lot of lipids, so there's a lot of ways in which you can take glucose and through lipogenesis, transform it into triglycerides. So it does store lipids largely as triglycerides, but it also can not only store steroids like cholesterol, but it can make cholesterol. It's also used to inactivate, as it were, the steroid hormones, and so break them down. Catabolically, again, gluconeogenesis, and it says in the table here, it's a primary site for beta oxidation that breaks the fatty acids down into acetyl-CoA. I'm not sure if it takes some of those fatty acids from the blood to help you break those down into acetyl-CoA and get rid of them. I'm not sure, but and what else? Makes ketone bodies from fat. I think that's about it. We also looked at protein metabolism, so transamination to make non-essential fatty acids. They could be made from essential ones or from non-essential ones. You probably wouldn't make it from essential, but I don't know that. Maybe in nutrition, you can ask. And then it does gluconeogenesis with it. Again, hormones can influence that. So what else does it have in here? It makes urea from ammonia. That's an important function since ammonia is a reactive molecule and you don't want to allow it to accumulate in the blood. It even mentions in their table that an inability to do that because of liver malfunction, like if your liver's turned into scar tissue and you have cirrhosis and you don't have enough liver tissue then ammonia can build up in the blood, and that's one of the toxicities that happens with hepatitis and cirrhosis. Synthesizes plasma proteins. I missed that in the protein part, or in the synthesis part. You could put it in there. So albumin and the alpha and beta globulins are all made there. Transferrin, angiotensinogen, plasminogen, lots of the blood proteins, with the exception of gamma globulins, which of course are antibodies. Okay. All right, cleaning services. So here you have those specialized phagocytes, the Kufler fixed phagocytes, and there's lots of them in there, and they're going to be doing that both to remove foreign things that are absorbed by your digestive tract, but also to recycle red blood cells, because the liver plays a role in grabbing damaged red blood cells and breaking them down into heme and forming from the bilirubin, and from the bilirubin, it can remove that bilirubin from the blood, or the bilirubin made in your spleen or your bone marrow that are also recycling red blood cells, and use that to convert it into bile. So it helps you remove that bilirubin. Again, cirrhosis of the liver or hepatitis can lead to inability to do that, and then you end up with jaundice because of too much bilirubin. Recycling services, well, red blood cells is probably the main one. We didn't have EPO up in synthesis, but it does make EPO. It even makes, I believe it made another, I can't think of what it is now, another hormone. I guess in the cleaning, it also takes toxins and drugs out of the blood, and that may be either absorbed by your digestive tract or even just in general, and helps to either store them or get rid of them. Okay, good for this. I mean, you could try and make this as extensive as you want. This is a table. It looks pretty much identical, except I think I called cleaning service. They don't have that to the one in the book. I'm talking about 24.5. All right, so does that help with the rest of the liver? I didn't even mention immune here. You could put immune stuff on this table. They don't have it on theirs. You could add another column and, talk about complement, making complement, responding to macrophages, and making fever, pyrogens, prostaglandins that act as pyrogens. There's all sorts of things you could add to this table. This isn't everything. So you could add more in your studying for the comprehensive. Okay, so there aren't any of these. I have tried with Canvas tests to do this because I kind of like them, but unfortunately, I could, I guess, if I made them an essay. I tried to make them like fill in the blank or something, and it didn't work out, but there aren't any on there. But we could take this, and it's saying correct the false statement. So all these are false, and you're saying what to change about this. So glucagon targets the liver. It does target the liver. That's true. You know that. And it says to increase its uptake of glucose. You could just change uptake into release of glucose into the blood. Then that would be good. And I think that's where I tried to, change one word here, fill in the blank. It didn't work out very well with Canvas testing. So there aren't any of these type. Okay, excitation of the sympathetic opens the external rectal sphincter. Well, first of all, the external is skeletal muscle, so that's not going to be innervated by the autonomic nervous system. But even if you say internal sphincter, it doesn't open it, it closes it. So too much wrong there. So you'd have to change close and internal, while excitation of the parasympathetic causes the internal to open. And that is true, right? So you'd be changing two things here to make this right. You're changing this to, you could also just change one thing and say, no, you'd have to still change two. You can't make somatic and then closes. Okay, hormones released by enteroendocrine cells of the duodenum always slow the motility and gastric secretions of the stomach. So not all, if nothing else, not all the hormones do act on the stomach. Plus, there's intestinal gastrin, and gastrin would increase. So the always is a giveaway in that one. It's not always. There's most of them do. Okay, bile manufactured by the liver is identical to that released by the gallbladder, but the gallbladder's role is to concentrate the bile. So that's not true. So gastrin secretion leads to the opening of cardiac, pyloric, and ileocecal sphincters. It does help you open these last two, but the cardiac, you don't want it to open. When gastrin is high, that means you're digesting things in your stomach. It's getting very motile. You want to have this one closed. So this one must not be sensitive to gastrin. VIP increases smooth muscle. It decreases, it relaxes them, right? Okay. This is right. I'm not sure I'm going to walk through this with you unless you're really desperate, because this is right out of that figure in your lab manual. So who digests the disaccharide? You could choose anything. You could say maltase here, sucrase, lactase, the product then. And you could just say a monosaccharide. That's fine with me. You don't have to tell me specifically if it's maltose what the products are. Mechanism used to move it in, unless it's fructose, it's going to go by secondary active with sodium. Maybe I will walk through the table. And as fructose, it's going to go by facilitated diffusion. They're going to exit out by facilitated. Lipase products, monodiglycerides and glycerol, fatty acids. Mechanism to move, simple diffusion for most of them, and then mechanism to move it out, exocytosis of chylomicrons. You could put lots of different enzymes here, but since I have this as a amino acid, it has to be one of the brush border enzymes. So it can't be trypsin or chymotrypsin because their products are di or tripeptides. They're not amino acids. You can, it says name two here. So sodium secondary or H plus secondary active, and then facilitated diffusion. This is just saying what moves the water. Well, you have that sodium potassium pump in the basolateral membrane moving sodium through, but also you could talk about as all these nutrients are absorbed, and they are absorbed largely through secondary active with sodium, that moves, water follows. And this is the concept of having oral rehydration therapy, that you need a little sugar, a little salts, and those will help move the water. There is no paracellular in the duodenum. So you're always going transcellular, so you need that sodium to help water move. If people had written here aquaporin, I'd probably allow that, but that wasn't what I was looking for. Okay. All right, so let's see, described on the line the stomach. So I changed this one, right? But we could kind of go through that. So long reflexes are coming from the central nervous system. And one of the things that Sana found or maybe it was Jessica, in my comments, and I haven't had a chance since we went right up to the wire to edit this, But in one of my comments, I said something about short reflexes with the cephalic phase, and there aren't any short reflexes. Cephalic is all long reflexes. I said something about they're always excitatory, I think. And that's true, that long, I can't remember. I'd have to look at the comment again. I don't have it open anymore, but I'll edit it. All the cephalic, though, are long. They're not short. I want to just make that clear. There are short reflexes that then can induce long reflexes, but those are going to occur in the gastric phase. So if you're talking about a long reflex, the only neurotransmitter that's excitatory that I probably in this question should have said during normal digestion, not the inhibitory side. The only long reflex neurotransmitter we talked about was acetylcholine. When you're comparing and contrasting, that would certainly be a similarity. But the amount of acetylcholine released is different. Long reflexes and short reflexes in the gastric phase increase the amount as you go through the gastric phase. If you're in the cephalic phase, you're only getting a little bit. This doesn't actually ask that. I guess I misread the question. So you're going to use acetylcholine for both of them in the gastric phase. So what's another similarity? A stretch, I suppose, is important for both. So stretch receptors go through the vagovagal reflex to increase long in the gastric phase, and they also work locally to increase short reflexes. Okay, differences. Well, there you can use the idea that one requires the central nervous system, the other one is using the plexes, the ganglia that are parasympathetic largely in the plexes. And differences, chemoreceptors that detect nutrients and pH mostly affect especially the nutrient one, mostly are affecting the enteroendocrine cells, causing that, and that's short reflex only.
Professor,
Yeah.
for the chemoreceptors, I may have misunderstood this before, but did you say that they also send like taste sensory as well?
I think what I said is that they are now considered similar to taste buds, but I don't know that they send any of that. Well, yeah, I don't think they send any signals, like sensory signals, up to the central nervous system. I don't think it's like you're actually, oh, wow, in my stomach going, that's sweet, and realizing that. I think it's just that they are structurally taste buds, not that you're actually getting a sensation of taste. Is that what you're asking?
Yeah, and maybe I wrote something incorrect in my notes.
Incorrectly. No, I don't think that they do, but the breakdown products from digestion, like glucose and such, do reach your hypothalamus and your hunger satiety center, but that's after the duodenum. I don't think in the stomach there's any sensation that goes up to higher centers, but I could be wrong about that, but I don't think so.
So is the vagovagal just to do with stretch then?
Yes, as far as I know, yeah. It may be pH too, but they don't really say, if I remember. Does anyone remember, since I haven't read it as recently as you guys? If pH is also sent centrally, because there's got to be something, I think it's local reflex though, when the pH drops below 2. I think that's all a local reflex, but somehow it must at least get the sympathetic involved, right? Because you're going to cause an inhibition of the secretion of HCl. And I think that's a local reflex, short reflex, but I could be wrong about that. Okay. So I would spend time with these phases. This seems now a little while ago, but ideally what I would do is work with each of the phases individually, the cephalic and gastric, but then now integrate what you know of the intestinal, since what I did in the lecture recording, and then even when we went through the lecture questions, is we didn't do the intestinal till well into the after we talked about structure of the duodenum and the jejunum and all, we went back and talked about the hormones and their effects in the stomach. We kind of just gave a blanket thing. But now you've got the table, whatever it is, 23, the one with all the hormones. And you might want to try and sew those together and bring that intestinal phase in to how they're acting on the parietal cells and the chief cells and motility, if that makes sense. To me, it's always good if you then later integrate stuff, since the book sometimes does it piecemeal and things are inserted in between. So this is that table. I'm not going to go through them unless there's some particular question you have. All you have to do is look at that table 23.1 and you can come up with 5. So what 5? CCK, GIP, gastrin, histamine. You could even, if you wanted to, have intestinal gastrin be different because it's on there. Motilin, secretin, I guess VIP is a neurotransmitter. Serotonin, if you wanted. We didn't talk about it much. There's a lot more than 5. And this doesn't say from the stomach or duodenum or anything. So it's wide open. Okay, so does anyone need me to do this first part? The 5 different types of cells? Because that was on the lab test. I mean, we could just name them, right? The surface epithelia that are in all 3 regions. This came up in our discussion this morning. All 3 regions of the stomach, gastric, fundus, body, pyloric, they all make an alkaline mucus because you're going to need to line the whole stomach with this thick mucus that protects the stomach, which goes to this last part. That's one of the ways the stomach is protecting itself. The chyme will be everywhere, so you better have that everywhere. But then when you get into the body, you have these acidic neck mucus producing cells that are making an acidic mucus. I don't know that those are in the other parts of the stomach. What we were talking about this morning was that if you had a chance to look at the 3 sections of the stomach in the slide that we own, what you would see is the pits become very shallow in the other areas in the cardiac and the pyloric regions. So my guess is they're shallow because you're not having parietal cells, mucous neck cells, and the chief cells in those regions. So that's the second one. Third one would be the parietal cells making HCl, but also making intrinsic factor and secreting that into the lumen. Then you have chief cells. Chief cells are making pepsinogen. And we don't know because they don't say, but my guess is they probably make the gastric lipase and rennet. So the other enzymes that are secreted, but they don't actually say that. It just, sort of makes sense to me that it would be them making it since they make pepsinogen, but I don't know. And then you typically, I guess you have stem cells there next, but again, I didn't stress that. The book does talk about it. And then finally you have, deepest in the pits, you have the enteroendocrine cells. So they could be close to the capillaries of more lamina propria than you see up in the areas where there's the parietal cells, and release their hormones into the blood. Okay, so if you take the parietal cell and you draw it out, the only change is, there are only a few things that are different between type A cells and parietal cells. And one of them is their sensitivity to gastrin, ACH, and histamine. And all 3 of these acting through that second messenger cyclic AMP system will upregulate the carbonic anhydrase and make more HCl. Whereas in the type A cell, when you have a lot of HCl or very low pH, you're going to upregulate the carbonic anhydrase. If anything, when the pH gets quite low, it gets downregulated in the parietal cell. So hopefully you can walk through that mechanism. If you need me to, let me know. So ways the stomach protects itself. Well, we said the alkaline mucus. There are lots and lots of tight junctions everywhere along the alimentary canal. So there's no paracellular. You don't want those juices to leak to the deeper tissues, or you'll have an ulcer. Another way they do it is by secreting pepsinogen in an inactive form, so it can't digest the carriers in the cell itself. They secrete H separated from Cl. I don't know if that put that in the list, but H plus is always secreted. I mean, it would always be separated from Cl, but it's not actually having little hydrochloric acid vesicles that it's releasing, which would be, I guess, one way it could do it. It doesn't do that. It protects itself not from damage, but from allowing its function by being highly mitotic, having its own stem cell population that renews the lining every 3 to 5 days. Is there anything I've forgotten? So again, if the short answer says list these, then all you have to do is list them. But if it says describe, then that means you have to give me what I just did, what they are doing, not just here they are, here are the 5 things. You'd get half credit for that. But if it's saying describe, now if it's saying list 3 ways, then you could write tight junctions. Many tight junctions probably would be better. Something like that, and you'd get away with it. Okay. Phew. So what time have we got here? Well, so we still have another 20 minutes. We could, and then did I set up I think I did set up where's my Zoom? Here it is. I think I set up another office hour after this, but if I didn't, I will now. Because I can't do the one later this afternoon. No, I didn't. So I will set one up and we can switch over to that office hour one. So because I didn't set it up yet, we can go past the 10:55 that I set this one up. Let me stop sharing and do some review. But I have a meeting starting at like 11:30, so we can't go past 11:30. So let me end this. Oh, not end this, but I stop sharing. Let's see if there is more stuff in here. So Alexa says the pH is stimulatory for G cells. That could be, if that's what the text is saying. And then, Noah's saying if you get less than 2. So it seems like it's a totally local, there's no sympathetic involved. I think that's not very clear in that figure, and that's what I'm remembering. So enteroendocrine, apparently, that's not something that I talked about, that enteroendocrine are deeper than stem cells. So I wouldn't test you on that. But someone pointed it out in the text that the stem cells are above the enteroendocrine. So the stem cells are apparently replacing those without replacing the enteroendocrine. I don't know what rising pH means. Okay. So here's another question. I'm sharing it because it's probably useful for others. For the defecation reflex, the internal sphincter will always contract from the parasympathetic. No, parasympathetic allows you to defecate. So stretch in the rectum is going to excite your parasympathetic. that causes contraction of the smooth muscle along the anal canal, but it must relax the sphincter, right, to allow you to defecate. So what's stopping defecation is the sympathetic activation causing contraction of that internal sphincter, and your somatic causing contraction of the external sphincter. Protein digestion in the mouth, there isn't any. There's no proteases released in saliva, if that's what people are asked. Okay. All right, so no proteases in saliva, but you have some lipase. It's not clear that much lipid could be digested because of no emulsification action. And then amylase is in saliva. Not clear how much you can digest that because you tend to swallow the food. And my guess is the amylase is inactivated once you get it in the stomach. Okay, any other questions in here? Let me just give you a little time and then. There are 2 short reflexes in the gastric phase, correct? 2, what are the ones why don't you tell me which 2 you're thinking of? Because, it depends on how you cut them up. So why don't you type in the short reflexes? Enteroendocrine and the other reinforcing the long reflex. The motility also increases due to increased local parasympathetics. Remember, the ganglia are on these organs, and that plexus can be excited by the stretch. It can be excited by just by the stretch that I can think of, but also by the enteroendocrine hormones released, like motilin or gastrin. And those are exciting the smooth muscle, but the ACH is also, and that would all be considered short if that's what you're getting at. And then the other is reinforcing the long, the vagovagal. Would be a long reflex, right? Vagovagal. So enteroendocrine by chemoreceptors would be short, enteroendocrine by stretch causing local increase of excitation and ACH. That would be the 2 maybe that you're thinking of as short. Okay. Oops, I forgot one question because we were put it off. And that's going back to the cardiovascular system. So this is a big question. What she's asking about is comparing and contrasting exercise to low blood volume on cardiac output. So other people can think of things that we can cover. That would be good too, but we can spend a little bit of time on this one because it's a big question. Okay, let me get chat open again too so I can remind myself of the question. Come on, Guy, open it. There it is. Okay. Okay. So what we're trying to do with this one is we're trying to have short-term effects. Let's do exercise. Really can't write today. That's going to lead to increased sympathetic. And that sympathetic is going to go off and make a couple targets. It's going to increase your heart rate. And it's going to increase calcium in the contractile. These work. Which will be stronger contraction, which will lead to increased blood pressure. And then the other main thing you're going to do is you're going to vasoconstrict. And that's going to lead to shunting, which will increase end diastolic volume. And that leads to stronger contraction. I'll put preload here. So all of that's going to go back and increase blood pressure, and that's going to be a stronger increase in blood pressure, so make the arrow fatter. That's exercise. Now, if you are exercising, you're also going to cause increase in thoracic pump and muscular pump. That's going to lead to increased venous return, which will increase end diastolic volume and preload. And again, that's going to cause an increase in contractility. And it's both vasoconstriction over here, and this is going to increase venous return, which means you're going to have more blood to pump out of your heart. So instead of the normal, 70 mLs that'll come out, you'll get a greater amount out. And instead of the normal 120 coming back, you're going to get more than the 120 coming back. So all of that will increase cardiac output. So the only variable you asked me about is end systolic volume, which is going to, the percentage-wise at least, is going to decrease whenever your contractility is stronger. Milliliters-wise, we don't know, because increased contractility, but you've increased venous return. So you're not at 120 anymore, you're higher. Your favorite preload, yes, that's right. Okay, so that's exercise. So then you wanted to do low blood volume. Well, right away with low blood volume, that's causing a decrease preload, which leads to decreased contractility. And we won't add numbers here because we don't know how much it's dropped, but it does lead to an increase in end systolic volume and a decrease in stroke volume. So your cardiac output goes down. So when you go to compensate for this, you're going to do two things. One, you're going to increase sympathetic, and then I'll just say see above, except, so everything we did above, except you're going to decrease milking by the skeletal muscles. You're not going to feel good, so you're not going to be moving. So you're not going to be milking. You may be milking less than you would normally, because you might be lying, really out of it if it's low enough. So you're not moving at all. Thoracic pump, though, may be increasing. So, but increase thoracic pump as you try to increase venous return. So if you can go up to this earlier one and see, all of these things, your heart rate's going to go up, you're going to increase contractility, you're going to vasoconstrict with your sympathetic nervous system. But the only thing that may be different is this one. In addition, though, you're going to increase renin release, which will cause increased vasoconstriction and more global vasoconstriction. So areas like skeletal muscles that might try and isolate from the sympathetic and override that won't. You're going to increase vasoconstriction to increase venous return again. You're going to increase aldosterone, and you're going to increase ADH. And these things are going to cause an increase in blood volume, which you didn't try to do with the other So in the exercise, I didn't put it up here. It does actually target the granular cells. So you're going to see a small increase in renin. But since your blood volume is not, your blood pressure is going up above normal, that may be obliterated. So even though the sympathetic targets it, but an increased mean arterial pressure may mean that there's constriction going on there already, and renin increase is not detectable because you have greater blood pressure on those granular cells as well. So this is really, I'm going to, just to simplify it, say that's really going to be unique to the low blood volume one. And these certainly would be significant for the low blood volume one to try and bring your blood volume back. I hope that's what you wanted. Okay, short term was both sympathetic and renin. Yeah, but I'm going to argue maybe in short term, renin is so insignificant since the blood vessels that they're riding on are getting initially stretched, which would, due to increase in blood pressure. But they're also being constricted a bit by the sympathetic. So that movement may mean that you're not seeing any real renin release. So renin is really mostly significant in the low blood volume. Okay, other things for our, okay, nobody's puts anything in there, so you're either overwhelmed by the stuff we did earlier, excuse me, or you don't know what to do with yourself right now. I would suggest for I talked about this before, for the comprehensive, I'm not sure what you guys are seeing in the comprehensive. I haven't published it yet because it's not totally finalized. Are you seeing any descriptors for it? So at this point, what I've written, I believe what is going to be the comprehensive will be 4 essay questions, so they're 10-point ones. So I would plan on that. And what I tried to do is spread it out over the course. And I tried to give you some choices so that you can, some of them, not all of them, but some of them, you can choose what body system you want to use to discuss that particular. So if you feel like, say, I'm a lot stronger in my cardiovascular system than I am in the kidney, you may be able to choose that cardiovascular system, and somebody else may be choosing the kidney. So the question will focus you. I told you this before. Now, 2 of them, I believe, are directly, about a certain body system. So I think 2 of them are like these choice questions, and the other 2 are more, you have to know something about the body systems. And I would use your essays I did try to base them on essays that we already had. So I'd use your essays and what you've written in your essays to be leaping off places to study for that comprehensive part. So because there are 4 10-point ones, I know you have 2 hours. Let's see how many people are here right now. I have to move this up. There's 19. So most of you, but not everybody's here. One of the things that I'm offering, and I'll do this on announcements, because I don't know you guys' test, but I've set it up so that I'll be there and at 10 of, it starts, or 10 of 8. And I think I made it a 2 hour and 2.5 hour time. So if it seems like time becomes really limited, and if everybody agrees, that means I get no feedback in the next 2 days that it's not possible, then I will try and make the time frame, say, 2 hours and 15 minutes. I think it'd be safe to do that because the way the final exam schedule is made is there's always a 15-minute break. And that way you have a little extra time if you feel like you need it. The only negative would be if somebody says, I have an exam at 8:30 or at 10:30, and I don't want to be writing this exam right up to the minute when the next exam starts. So if they really object to it, I can't do it. But at this point, I think I've set it up so that there's a little 15-minute thing. I also don't have quite as many essays in the midterm part. I think there's 2 10-point and I forget 2 or something 5.1s. But I'll post that probably today as I finish writing the test. The rest are multiple choice, and then I have some that are kind of a fill in the blank, but you don't have to, they're going to be in the essay format. So that way, when you go to fill in the blank, I'll just be reading those. So, okay. So let's wind this up at 11, unless somebody has, I have a few more minutes.
Dr. Kaufman,
Yeah.
going back to what you said about the vagal reflex, this figure in the book on page 875, it says that chemoreceptors as well as mechanoreceptors can detect internal stimuli and then send signals to the central nervous system. So
Osmo or mechano to the local. Yeah, it's pretty simplistic because then if you go and you look at the more detailed figure of the gastric phase, I believe, where is it now? Here it is. You see, if you look at the figure 2319 on 890, it's only showing long reflexes from stomach distension of stretch receptors, and then food chemicals, pH and rising pH and activate chemoreceptors. Those are only going to the enteroendocrine. They don't even show it going to the short ones, which, as far as I know, they don't. So what does it say in the stomach distension activates stretch receptors, initiates strong, short and long. Chemical stimuli directly activate gastrin cells. Gastrin plays a role, blah, blah, blah. The proteins in the food, the pH stimulate gastrin secretion. Yeah, so they don't really talk about that.
I think the reason that, I think in the lecture you had drawn a line from 2B up to 2A to show that it wasn't in the part of the figure, but I remember.
Yeah, and I think the line was to go to short reflexes because my understanding is that that's a short reflex.
Okay.
Not to go to making more long reflexes. I don't see anything in here about it going up to, yeah, so there being anything about chemoreceptors sending information centrally and becoming part of that vagovagal. Let's see where it actually talks about the vagovagal. I mean, if you go online and you find something different, we're just going to go by what the book limits it to. So vagal fibers, where does it directly talk about vagovagal? Things are always more complex than they're going to have in the book. So unless it's important, I'm not going to.
I think it's on page 890.
It is on 890, I must have. But do they actually describe? Yeah, because I read all of 890. I mean, it's kind of referring to it, but it's not really defining it anywhere, does it?
Mhm.
It just does it in that figure, says vagal. Huh. And that's the only place in the index? Okay, I think we're going to have to go with that. Was the wrong thing here. Okay, that's my understanding is that the food and the chemicals, I'm using that figure 2319 is largely, and the text is largely stimulating just a short reflex through the anterior endocrine, but they also, as far as I know, well, we're not sure. If I made another arrow, I'd have to look it up, but it would still be short. We're not going to do any long. It may also go through that plexus and stimulate more parasympathetic, but that I'd have to Google to figure out.
Okay, thank you.
Okay. Any other questions? All right, I am going to switch over for the next 20 minutes to setting up let me set it up, a office hour. I wanted to see when we got done with this one. So if you have any individual issues and you want to talk to me about individual issues, let me set it up right now. Quick launch. You can, and we'll do it for, what time is it now? We'll do it for 30 minutes. You can do that. Switch over to my office hour, Shell, and you can ask me. So I'm going to leave this meeting now and then be over there. And I will otherwise see you you can show up anywhere from 10 of 8 on Thursday for the test. And then I'll keep it open until, it's supposed to be done at 10. I'll keep it open until 10:15 unless I hear otherwise. So that means I'll change the outline of when the test closes it automatically to 10:15 unless I hear from somebody that they really can't do that. Okay, I'll see you over an office hour. Otherwise, happy studying. See you on Thursday. Bye.